And let's welcome Lucy Williams from the University of North Carolina at Chapel Hill, who will tell us more about MeCP2 biology.
First, I'd obviously like to thank the organizers for giving me the opportunity to discuss a project that is really just under development, only about a year old. And as just mentioned, it focuses on MeCP2, its dysfunction in relationship to RETT syndrome. So as Monica discussed, methyl CpG binding protein 2 reads modified cytosines and is involved in fine-tuning transcription, particularly through temporary repression. It is ubiquitously expressed, but it is remarkably high in the brain, where it's thought to be on the order of 1 MeCP2 molecule per every 2 nucleosomes. In hemizygous males, there's a complete loss of MeCP2. It's lethal. And in females, as just discussed, dysfunction of MeCP2 causes RETT syndrome, which Monica discussed the broad severity of symptoms in her introduction. What I want to point out particularly is that RETT mouse models do recapitulate the disease. However, there are some problems, and the symptoms are actually much less severe in mouse, both in the severity of the symptoms and also the timing of the symptoms, particularly in females. So as Monica discussed, in their assay, they're actually using male RETT models instead of females. And there are 2 categories of treatment strategies that are under development right now for RETT syndrome. The first is to target signaling pathways, for instance, that are downstream of MeCP2 and to ameliorate the symptoms, for example, like GABA, or we can target MeCP2 itself through, for example, gene therapy, where we can reintroduce MeCP2 transcripts or protein back into the brain. So this second strategy is extremely attractive because it offers the opportunity to cure RETT syndrome. However, the situation is obviously complicated because MeCP2 is an X-linked gene. So in the brain of RETT girls, we have approximately a 50% ratio of cells that have inactivated the mutant copy of MeCP2, and so those are phenotypically wild-type cells, and then the cells that are now expressing the dysfunctional MeCP2 copy, so those are the dysfunctional MeCP2 cells. So this sets up a situation where we can't just reintroduce an abundance of transcript into the brain, because we would then overexpress MeCP2 in those cells that are expressing the wild-type copy. This is particularly a problem because an overabundance of MeCP2 is associated with MeCP2 duplication syndrome, which is a neurological disorder which shares the clinical phenotypes of RETT syndrome. So then what we're looking for is basically is there a narrow window in which we need to express MeCP2 to get a therapeutic benefit without extending beyond those thresholds and having a consequence that could actually make these girls worse. So then the basic questions are, first, well, how variable is MeCP2 within just a normal population? And do steady-state mRNA levels actually correlate with protein levels? To address these questions, I am using a systems genetic approach, and I won't go into the details for this talk in the interest of time, but I will be using the collaborative cross inbred strains, as well as the diversity outbred mouse populations. And the important point to note is that these are coming from the shared 8 founders, and so we can integrate data sets from these 2 populations over space and time. So to begin to first address what level of variability could exist for MeCP2 expression, I isolated total RNA from whole brain of 123 diversity outbred females. So these were adult females. They were over a year and a half age. They had actually given multiple litters. So overall, they looked fairly healthy. And in this case, what we found was remarkable. And then we have over a 60-fold level difference across the population in MeCP2 expression level, and in a continuous level of variation. Again, this was remarkable because the model suggested that there would be a very narrow window of MeCP2 in the brain. Now, that was a qPCR assay, so we went to validate the qPCR assay by analyzing available RNA-seq data sets. So this was, these RNA-seq data sets were from total RNA isolated from the forebrain, now of F1 hybrid animals of crosses of different inbred CC strains. These animals were actually of a different age. These were much younger, between 9 and 12 weeks of age. I had 27 different hybrid lines to look at, but interestingly, we saw similar levels of variability. So here in the red triangles, I'm showing the median expression value. The black dots are showing replicates within the CC strains. So we can see broad levels of expression variation from individual F1 line to F1 line, and also that within the F1 lines, this variation is heritable. Okay, so from there, I then went into the inbred CC strains. Because these are reproducible, I can begin to do molecular biology experiments on these in the future. And I selected at random 27 CC strains. The box plots are representative of 8 or more adult females from every line. They are color-coded according to the shared MeCP2 haplotype from the parental founder. So you can see that we have expression variability across MeCP2 haplotypes or within MeCP2 haplotypes. In addition, I'm showing the parental founders, WSB, Castaneous, and PWK. So those were the most variable founders for MeCP2. And you can see that the expression in the individual CC strains extends beyond the expression level for those 3 founders, suggesting that in addition to cis-level variation, trans factors are modifying MeCP2 expression levels in this population. So what I've shown you so far is expression level, which could be obviously quite variable and not translate to protein level variability. To begin to address this question, I took a simple approach first, and I chose lines that were remarkably low, remarkably high, and average, just to begin to test protein level. And so far, what we found remarkably is that at least in the adult brain, the protein level appears to be correlating with the RNA level. There are some outlier strains, and I think we would expect this, and those outlier strains by themselves could be interesting to study in the future, to specifically look at why those lines are different. But I think this suggests that we can now begin to look at eQTL and protein QTL level analyses in the animals. So then, just in summary, then what we've shown is that there's this remarkably high variability from individual to individual. The question is, does this translate to human? If it does, could MeCP2 itself be functioning as a modifier of the phenotype, particularly in the girls that have a mosaic expression of MeCP2 in the brain? We've shown that the levels are under genetic control, and so we're setting the stage again for molecular studies, as well as to be able to link phenotype. Because now the important question is, if we have this broad level of MeCP2 across these lines, are these animals phenotypically normal, or do we have lines that have mild expression and also show phenotype, and is that the expression threshold that we need to reach? So, as I said in the beginning, the female mouse in particularly is not the best model because the symptoms are much less severe. This makes behavioral assays very difficult, especially in my situation where we may have a mild underexpression or a mild overexpression, not just in knockout. So I wanted to start to sort of play with other assays which we might be able to utilize. So in this case, we're looking at seizures. And so 50 to 80% of RETT patients develop epilepsy with diverse seizure phenotypes ranging from myoclonic to generalized. But natural history studies indicate that there's really no correlation between the severity of the mutation in MeCP2 and the severity and the seizure, or the type of seizures that the girls display. But that being said, in mouse, mild overexpression and underexpression has been reported to cause a heightened sensitivity to seizures. So I'm using an assay where we are exposing the animals to a putative GABA antagonist. So in this situation, every animal will get a seizure. What we're looking for is the latency to a general seizure threshold, and that gives us a measure for seizure susceptibility. So now looking at the line at 16 CC lines, again chosen at random, with B6 and DBA as our controls, we can see that we have remarkably tight data, so shown just in the individual dots. We can also see that the lines are spread out for their seizure susceptibility, and interestingly, a line that has shown, so CC58 shown in green, has shown to have low protein levels, as well as other neurological phenotypes, appears to be the least susceptible to seizures in this case, directly contradicting the previous mouse studies. So the question is this just a situation where MeCP2 isn't reduced enough, or do we perhaps have a modifier that could be overcoming the effects of MeCP2 loss in this situation? And so these are directions that we're moving forward in the future. The other thing of interest, which isn't related to MeCP2 but came out of this screen, is that we have 3 lines whose data are shown in red that actually had a phasal response in the naive trial. So the first response to fluorothiol in this case gave 100% lethality, for example, in CC8. So we think we may also have uncovered new models for SUDEP, for sudden death and epilepsy, and we're also now investigating that in the future. So then I want to thank, of course, Fernando and the other members of the UNC Systems of Genetics Core, Darla and Ginger, for help with mouse breeding, or really basically doing my mouse breeding for me, as well as Marty Ferris for support, members of the Fernando's lab, Andrew and Rachel, for help with dissections and mouse breeding and whatnot, members of the Terry Magnuson's lab, and also the seizure susceptibility studies are in a collaboration with Ben Philpott at the UNC Neuroscience Center. And the RNA-seq data sets that were analyzed are part of the maternal nutrition studies with Fernando, Will Valdar, and Lisa Tarantino in the SEGS Consortium.
We have time for a few questions.
Yeah, when you mentioned the MeCP2 mutation has a weak phenotype on a mouse, is that one background, or has that mutation been put into a variety of backgrounds?
Sure, I should have mentioned that. The number of backgrounds it's been put into is relatively limited. So I think C57 Black 6, FBB 129, but the classical laboratory inbred strains, yeah.
So. Hi. Sorry. I'm sure you've thought about this, but it's, I know, very difficult to wait for spontaneous seizures, but as soon as you add an inducer, then you raise the possibility that the strains vary in their metabolism of that chemical.
Yes, yeah. So, right, we're looking into that. It's just basically in the very beginning stages. We would have to look at how it metabolizes. In the SUDEP models, we need to look at heart defects and other potential complications that may come up. In this situation, the fluorothiol is breathed in by the animals and basically immediately metabolized. So, but we would need to, for example, look at different concentrations and all of that, absolutely.
Your susceptibility to fluorethyl's, so
yes,
they may be varying in your strain also.
And of course the sodium ion channels would be the first thing that we would look at. And at least in those lines, I've looked at all the known epilepsy susceptibility genes, and I don't see anything coming out immediately from those known genes or pathways. Yeah.
Hi, great talk. Have you thought about crossing in the MeCP2 mutation into all the CC lines so you can directly test the effect of the alleles?
I've thought about it. I don't have that much funding to do that. I would love to do that.
Just one cross.
Yeah, it would be, I mean, the idea would be to cross it into the remarkably low and remarkably high lines. The other idea we have is we're testing out with Terry Magnuson's group a CRISPR approach that we could perhaps try that could potentially be faster, but those are all, I think the first thing to do is just to assess the phenotype in the lines themselves before we put into the situation and knockout, just because of time and funding, honestly.
Thank you, Lucy.